COMT, or COMT. COMT stands for Catechol-O-methyltransferase. It catalyzes the omethylation and thereby inactivation of catecholamine neurotransmitters such as dopamine, epinephrine, and norepinephrine, and catechol hormones such as estrogen. COMT shortens the biological half-lives of the following neuroactive drugs: L-DOPA, alpha-methyl-DOPA, and isoproterenol. The catalytic activity for COMT is S-adenosyl L-methionine plus a catechol. The cofactor is magnesium. The molecular functions for COMT are Catechol-O-methyltransferase activity, which is a catalysis of the reaction S-adenosyl L-methionine plus a catechol, which equals S-adenosyl L-homocysteine plus a glycol. O-methyltransferase activity, which catalysis of the transfer of a methyl group to the oxygen atom of an acceptor molecule. And magnesium ion binding, which interacts selectively and non-covalently with magnesium ions. The biological processes are cellular response to phosphate starvation, dopamine catabolic process, female pregnancy, methylation, negative regulation of dopamine metabolic process, negative regulation of smooth muscle cell proliferation, neurotransmitter catabolic process, regulation of sensory perception of pain, response to lipopolysaccharide, response to pain, small molecule metabolic process, xenometabolic process, developmental process, estrogen metabolic process, learning, multicellular organismal reproductive process, negative regulation of renal sodium excretion, neurotransmitter biosynthetic process, positive regulation of a homocysteine metabolic process, response to drugs, response to organic cyclic compound, short-term memory and synaptic transmission. COMT V158M and COMT H62H result in 3 to 4-fold difference in COMT enzyme activity and may contribute to the process of mental disorders such as bipolar disorder and alcoholism. COMT is related to cisplatin toxicity. Cisplatin is used to treat cancers of the testicles, ovaries, or bladder. People with the variants V158M and H62H homozygous can have trouble breaking down dopamine, epinephrine, norepinephrine, and estrogen when they are homozygous on the variant report and COMT is not functioning properly. If your practitioner sees that you have COMT V158M and H62H homozygous, he or she may want to check your neurotransmitters, adrenals, and sex hormones, especially if there's a history of PTSD, panic attacks, anxiety, schizophrenia, autism, and hormone-related cancers. Sterling Hill Airday from MTHFR Support has spoken with many psychiatrists who have run variant reports, and several have reported when a patient is chronically ill and has COMT V158M and H62H green on the report, most of the time these individuals have lower levels of dopamine, epinephrine, and norepinephrine. When psychiatrists have seen someone heterozygous for these mutations and the patient is chronically ill, they can be low or high on catecholamines. When patients are homozygous for these mutations and are chronically ill, they normally see an elevation in catecholamines and estrogen. Through some amazing doctors like Richard Death, PhD, and Amy Yasko, PhD, we have learned that methyl B12 may not be the best form of B12 to people with COMT, V158M, and H62H homozygous and heterozygous. Too many methyl donors can have a negative reaction on these individuals. There are two types of COMT activity in the body. The long form is called membrane-bound Catechol-O-methyltransferase, or MBCOMT. It is mainly produced by nerve cells in the brain. The shorter form of the enzyme is called soluble Catechol-O-methyltransferase, or SCOMT. This form of the enzyme helps control the levels of certain hormones. Other tissues, including the liver, kidneys, and blood, produce a shorter form of this enzyme. Catechol-O-methyltransferase is important in the prefrontal cortex, which organizes and coordinates information from other parts of the brain. This region is involved with personality, planning, inhibition of behaviors, abstract thinking, emotion, and working memory, also known as short-term memory. COMT helps the prefrontal lobe function with catecholamines. 22q11.2 deletion syndrome is a deletion of a small part of chromosome 22 where COMT resides. People with this deletion are more prone to mental illnesses such as schizophrenia, panic disorder, anxiety, PTSD, and bipolar disorder. Remember, if you have any concerns about the COMT gene, please contact a practitioner who is familiar with epigenetics and nutrigenomics.